Hi, this is Botany 101, General Botany. All right, today we're going to do session 3. We're going to do the first part of the plant cell. For today's objectives, okay, we're going to include what is the cell theory, all right, because today we're actually talking about the plant cell and we have to talk about the cell theory, yeah, because that does have to do with the cell. Next, we're also going to talk about early uses of the microscope because generally cells are microscopic, all right? So in order to observe most cells, you have to use a tool called the microscope. Thirdly, okay, we just talked about the types of tools that we use. So what are the types of microscopes, okay, that we use to investigate the plant cell? Fourthly, what are the components of the plant cell? Because a plant cell is a relatively small unit, but it is made up of even smaller components. Okay, similar to how molecules are made out of atoms. Yeah, remember we had the talk about atoms last time? Yeah, and subatomic particles and etc. Finally, what are the functions of these parts? Okay, so one of the things we have to establish, do you recall when we were talking about the different kinds of life and talking about the different components and different properties of living things? Well, one of the things we talked about was that generally all living things are cellular. Okay, they're all made up of either one or more cells, right? So let's see, plant cells are pretty small too. They're about maybe between 10 and 100 micrometers long. You see that U there? It's not supposed to be a U, it's supposed to be a Greek letter mu, and that stands for micro, yes, as a prefix to meter, okay? So plant cells are about 10 to 100 micrometers long, so you might think, what is a micrometer? You know, what is your point of reference? Well, if you can think of a meter, okay, a meter as being about 3 feet long, a meter is equivalent to about a yard, okay? 1 millimeter is 1/1000th of a micrometer. So it's really, really small, pretty small, okay? Usually you'll see it on a ruler. A micrometer is a millionth of a meter long. So you can tell from this that generally plant cells and all cells in general, okay, are very small. They're very small indeed. Okay, so because they're very small, we do need to use microscopes, okay, in order to look at them. Now let me talk a little bit about living things. Okay, so living things, part of the definition is they're made up of one or more cells. Okay, so we can think of the something like what is life, like we talked about last time. So we have living things made up of cells, and we also have other sorts of, you know, things that are not made up of cells. And among these, when you think about it, what is a virus? A virus, many of us might think of it as being something alive, but it is not actually alive. A virus pretty much is a piece of nucleic acid. Remember we talked about nucleic acids in the last module. So we talked about RNA and DNA. Remember those are the two different kinds of nucleic acids we talked about. So what a virus is pretty much is a piece of DNA or a piece of RNA encased by proteins. Okay, so that's actually the basic kind of virus when you think about it. And viruses then are not cellular because they do not have the components that make a cell. Okay, 3 main components of any cell include the cell membrane, also known as a plasma membrane. You also have genetic material. So viruses do have genetic material, but they do not have membranes. The third component of any cell, okay, is the cytoplasm, or the fluid that's inside of the cell. Okay, so viruses are considered by a lot of biologists not really to be alive. They're not considered to be living things, even though many of them are infectious agents, right, that can infect us, and also they infect plants as well. Another sort of item that comes up fairly recently is the idea of prions. Has anybody heard of prions? Yes? So what are they? Think about it. They're not cellular. What prions are, they're, some people can refer to them as rogue proteins. Okay, so remember proteins are another kind of macromolecule. It's another one of the 4 basic kind of biological molecules that we find, you know, in life. And generally, prion is just a protein. It is not cellular, it's not encased by a membrane, but prions are sort of scary. Anybody ever heard of mad cow disease? okay, or scrapie, which happens in sheeps. Well, these are diseases that are infectious that they're thinking maybe can cross into humans too. And these are caused by prions, okay, which are proteins. They're not cellular, so they're not really alive. But, some people say that you can actually, not kill them because they're not alive. Okay, so that's sort of like something to think about. So between something like a rock at one end and like a human being or a plant in the other end, non-life and life, there are actually intergradations between them. So just think about it, what is living, what is non-living, just something to think about, okay? So anyway, what we have here is a nice image of a plant cell. Okay, so we're going to go over some of the parts of the plant cell today, okay, but we're going to have a little bit of a detour talking about some of the tools that we use to study the plant cell. But first of all, I want you to look at this illustration, and you can see from the outside in, you have here, cell walls. So plant cells have cell walls, okay. Interior to the cell wall, you generally have this part that is yellow, the cell membrane, okay, also known as the plasma membrane. Interior to that, here this liquid that's kind of depicted here as a light blue color, okay, this liquid is called the cytoplasm. And in the cytoplasm, you'll have organelles. Okay, we'll go into some of these organelles in a little bit, all right? And then you also have, something called a central vacuole. You'll also have the nucleus, okay, which sometimes is the largest part inside of a plant cell. Okay, so we're going to go over these little by little, one by one, okay, as we progress. So we're going to start talking about the different organelles and different parts of the cell today, and we'll continue in the next session as well, okay? Now, in order to study the cell, remember that cells are really, really small. They're only like 10 to 100, right, micrometers or millions of meters, very, very small units. So we had the fact that not until the 17th century, until 1665 or into the 1600s, Robert Hooke was not exactly the first person to look at cells, but he was the first to actually, notice. Okay, so what he did is he looked at something called cork, which is also known as bark. Okay, so Robert Hooke in 1665. So I believe Van Leeuwenhoek was another person who looked at cells. So if we can actually look under the elmo here, what you can see here is this is a cross section, okay, of a woody plant. All right, what are the things you see here? For one thing, okay, we have here, this is the wood of the plant. Later on, we're going to find out that this wood is or was the water conduction part of the plant called the xylem. Don't worry about that right now. We're going to get into that in a couple of sessions. But outside of this woody area here, you have bark. Okay, bark is non-living tissue produced by a secondary meristem, which is called the cork cambium. So it is also known as cork. Okay? All right. So generally, Robert Hooke, okay, looked at this cork. Let me put something else under the elmo now. Okay. So we have here, a cross section of a plant, and here we have here bark from the paperbark plant, Melaleuca. Okay, so the bark of a plant is non-living, also known as cork. So it's not a living part of the plant. So what does it do? It protects the different parts of the plant that are interior to the bark. So generally, up to a certain point, you can peel bark off of a tree and it won't really hurt it, okay? Up to a certain point, but below the bark, you'll have living tissues, right? And in that way, generally, you might be able to hurt the plant, okay? So Robert Hooke, he actually looked at cork, okay, and he was a person who actually called these units that he saw under a microscope cells, okay, because what he saw, since cork is non-living tissue, he saw these small little components that look like empty boxes, because in the olden days, in the 1600s, you actually had people who went into the religious life in Europe, including monks and nuns, yes? So what do these people do? They take a vow of poverty among some of their, other vows that they take going into the religious life, and because of that, if they were lucky, maybe they had one small room to sleep in, maybe with a pallet on the floor, a place to hang up their clothing. That was about it. Maybe, their rosary and their book, the good book, a Bible. And other than that, since they were sworn to a vow of poverty, many of them didn't have much. So Robert Hooke looked at these little, little units making up the cork and saw that there wasn't anything in them, so they reminded him of monk cells or nun cells. So he called these components cells, okay? Right? Now furthermore, after the 1600s, 17th century, there wasn't much advancement in terms of optics. Of course, optics was a science that was developed, I believe, by Isaac Newton, before that. So between the 17th century and the 19th century, there wasn't that much further development in optics, because of course, microscopes, you generally need to have lenses to look through, right, in order to see something so small as a cell. So by the mid-1800s, there were these a botanist and a zoologist. Yeah, botanist studies plants, zoologist studies animals, and these two men were are known as Schleiden and Schwann. Okay, they worked in the mid-1800s, and optics had progressed to the point that you could actually see things in greater detail. Okay. So by the 1800s, by the mid-1800s, they had enough data to put together something that we now call the cell theory. So you might think, okay, what is the cell theory? The cell theory holds that the cell, okay, a single cell or more than one cell, the cell is the basic unit of life. Okay? It has all the qualities, in some cases, of everything it needs to live, okay, including various organelles and other, components inside of the cell. So the cell is a basic unit of life. Furthermore, all living things, all organisms are made up of one or more cells. So when you think about it, think about viruses. Remember, they're a nucleic acid and they are a protein coat. They don't have a cell membrane, okay? They don't have cytoplasm. They are not cellular. Therefore, most biologists would say that viruses are not alive. They're not living. That's why you can't kill them. Another thing that Schleiden and Schwann, another conclusion they came to, was that all cells arise only from pre-existing cells. Another way of saying this is that all life comes from life. You can't actually have living things coming from non-life. That isn't possible, at least not in today's environment and not in today's world. Okay? Another thing, too, that we must stress is that cells are microscopic, so people have used the microscope to observe cells, to look at their, substructures and etc. Okay, so cells are usually microscopic. There are a few exceptions to the rule, such as eggs. Okay, eggs, hen's eggs, ostrich eggs, which are pretty darn big, So eggs, of course, are single cells, but they're something you can see in a macro sort of proportions. Okay, so generally most cells are very, very small. Did you even know that there's even a bacterium which are almost all microscopic? There are just a couple that you can actually see with your unaided eye. There may be a couple, maybe about 1 millimeter large, very, very small. But still, there are a few bacteria that you can actually see without a microscope. But usually, as a general rule, cells are microscopic. That's what happens when you're taking an introductory class, and this is one reason why introductory classes are often very difficult, is because you're learning a whole new terminologies. You're learning a whole new, terms and descriptions of things that you're unfamiliar with. So if you're being bogged down and you're feeling a little burdened, don't worry, you'll get it. You just have to apply yourself and study. And so one of the things we have to say is that generally introductory classes are more difficult than higher junior and senior level classes, just because you're going very broad. Okay? Anyway, just remember, so one of the things you might find out about taking introductory courses is that you have to memorize the generalities, okay? And then you also have to memorize the exceptions. So generally, cells are microscopic, okay, generally, as a whole. Right? Now, microscopes, okay? One kind of tool that we use is called the compound light microscope, okay? The compound light microscope, okay, generally has more than one lens. That's why they call it compound light microscope. Because you know that when you have money in the bank or you have investments, some of us will actually get there one of these days maybe to have money, in the bank. Yeah, I know in Hawaii making a living is not easy. But a compound light microscope, okay, you will actually have different lenses that make it up, including one lens, okay, in one place and the other lens in another place, okay? So here we go, we have here an actual compound light microscope, all right? And then here we see that generally it looks like a kind of complicated, rather intimidating sort of machine, but it is a tool, just like any other tool. And we use a compound light microscope to study cells, okay, especially cells that are still alive. You can actually look at also prepared slides, and this is something that we'll be doing in the lab, okay? But right now I want to go over some of the parts of the microscope that you should know about. Okay. First of all, since we have different lenses in here, one set of lenses are in this part of the microscope, and these are called the eyepieces, also known as the oculars. And in here you have, generally in these student microscopes, you have a magnification of 10 times. Okay, so if you were to look at something under these lenses alone, it would magnify whatever you're looking at by 10 times. So if something was 1 millimeter large, generally what you'll do is that if you're looking at under 10 times, it's going to look like 1 centimeter large. It will actually give you the details, and 1 centimeter is about the width of your little finger. Okay, okay. So generally they're about 2.5 centimeters in an inch. All right. So here you have the oculars. Next, you have another set of lenses in these parts here. Okay, and these are called the objectives. And you can see that it will actually rotate from scanning power to low power to high power. Okay. Going back to scanning power, this particular one generally has a red line in it. You can't see it that well from here. But here you'll actually have a red line here that will tell you that this particular objective magnifies to 4 times. So how do you find the total magnification? You multiply the magnification of the ocular lens with the magnification of the objective lens. So you have 10 times in the ocular lens and you have 4 times in the objective lens. So what is the total of magnification if you're looking and you're looking under the microscope and using scanning power? Think about it, okay? 10 times, 4 times. Okay, so generally you'll have a total magnification of 40 times. Now if you were to actually turn this knurled knob here, okay, which is called the revolving nose piece of all things, and you look under 10 times, which has this yellow line here, all right, 10 times magnification under the ocular lens, 10 times magnification under the objective lens, what will be the total magnification? It will be 10 times. I'm sorry, 100 times, I got it wrong. Anyway, so it will be 100 times, yes? And then if you were to turn it further, okay, under this blue line, this will magnify, this particular objective will magnify for 40 times. So it will be 10 times 40, which of course is 400. Yes, I can do a little bit of math in my head. Anyway, if we were to look at the microscope further, you see this is a place here you can actually move. This is where you would put your specimen, okay, on a slide. Okay, on a microscope slide. You'd actually, I should have brought a slide, but here, this is called the stage. It's important to know this part, the stage. We also have other parts of the microscope, including these knobs here. Okay, so one of the things you'll do is that, you'll get your microscope out of the cabinet, you'll, unwind the cord, you'll plug it in, and then you make sure that this is all the way to the bottom, and you'll put your specimen in here, and then you use these knobs, okay, these knobs here to move the stage, side to side or back and forth. And then what you do is that you use this large knurled knob here to get it and looking in the microscope at the same time until you get it focused, okay? After you have it focused at scanning power, you will then change it to low power. The one with the yellow line on it, right? And then you'll use a small knob here. So we have the large knurled knob for scanning power. We have the smaller, coarse focus adjustment knob for scanning power, and fine focus knob, because what this fine focus will do, it'll only move the stage very slowly, so you don't have the possibility of sending the stage crashing into the lens. So I guess anybody who knows about optics, who has glasses, or actually does photography as a hobby or as a profession, knows that lenses are very expensive. And this, so generally with a microscope, you want to treat it with TLC, and also very respectfully, always use 2 hands whenever you're carrying it, okay? and make sure that the lens, because lenses can be very expensive, yes, they can run into hundreds of dollars. Okay, so we have to be careful with the equipment. Like this is a pretty fine instrument. It will actually, you know, stand up to a lot of use, okay? But at the same time, we have to kind of respect it as well, because this is a tool through which you're going to be observing plant cells, okay, in the lab. Right, let me put this back here. Alrighty. All right, then, so we talked about total magnification would be the magnification of the objective lens times the magnification of the ocular lens, yeah? Just like we just said, okay? Now, generally also, we have another tool. So we have compound microscopes that lets you look at something that is still alive. You can actually make a slide of a living thing and look at that under the microscope, and the plant or the alga or any other thing that you're looking at under the microscope can still be alive. Now we have another tool known as the electron microscope, okay? On the compound microscope, you will actually have magnifications maybe up to, what? Maybe about up to maybe almost 1,000 times, okay, almost 1,000 times magnification. But as you go up in magnification, you kind of lose a little bit of what they call resolution. What is resolution? The resolution is the ability to kind of look at two things that are close together and still see them as separate from each other. Okay, under high magnification, very high magnification, sometimes if you're looking at some things that are very small, it looks like instead of two things, it looks like one thing. Okay, so that's what they call resolution, the ability to resolve or see two things near to each other under the microscope. Okay, so that is a drawback of the compound microscope. Now the electron microscope, okay, has a good quality is that generally you can see things that are very, very, very small. You can even see atoms nowadays with some. And one kind of electron microscope is known as the scanning electron microscope, also known as SEM. We use SEM, okay, for scanning electron microscope. You also use SEM for scanning electron micrograph, which is an image that people take of cells or cell structures, okay, under the microscope. Okay, scanning electron micrograph. That will usually give you surface features of whatever you're looking at. All right, okay, so the SEM will give you what it looks like on the surface of what you're looking at. Secondly, we also have something called a TEM, also known as a transmission electron microscope. The transmission electron microscope is a tool under which you can take images of something with which you're actually looking at, where so electron microscopes, of course, uses electrons, whereas a compound light microscope uses light that you need to shine through a specimen. The electron microscopes, you need to actually beam or shoot a bunch of electrons, okay, at your specimen. Okay. One good thing about the electron microscope, you can see things at very, very, very tiny. Very tiny. You can actually get up to maybe up to 200,000 times magnification of what you're looking at. Okay, that's one very good thing about the electron microscopes. One of the things that is not so good is generally whatever you're looking at would tend to be non-living, because usually the specimen is held in a vacuum, and pretty much a cell and living things cannot live in a vacuum. Okay, that's a drawback. But here, okay, here we'll have an image, okay, of an electron microscope, and what you can see here is that it's a very, complicated thing. Usually you can see that it is so large that you usually have to sit at a console, okay, and then look through the viewfinders in here, okay, because it's very large, because it has to be able to generate the force to shoot a whole bunch of electrons. And remember, electrons are components of an atom, okay, with a negative charge, okay. Now here we have an image, okay, of bacterial cells. So this, I believe, is Escherichia coli, which is a coliform bacterium, and this is a bacterium that live in our gut. Okay, it is actually a symbiont. It lives with us, and it helps us to break down, food particles, helps us absorb, nutrients as well. So you can see this is an SEM, or a scanning electron micrograph. What you can see is surface features of these bacterial cells. It's remarkable. Look at that. Okay, so these bacterial cells, very, very small. So here you can see that these cells are on a scale of about 2 micrometers. This is actually the mu, yeah, that we talked about a little earlier when we were talking about micrometers, where plant cells are between usually 10 to 100 micrometers. But here you can see that these bacterial cells are only maybe up to 2 microns or 2 micrometers long. Very, very, very small, 2 millionths of a meter. large. All right, very good. Now we're going to get into parts of the cell, starting with the cell wall, okay? So generally the cell wall, all plant cells have cell walls. We animals do not have cell walls, okay? So if we look at me, I can move, yeah? Animal cells, we can move, so we cannot actually utilize the cell wall because we need to move. We humans, and as well as other animals, have evolved either methods by which to chase after prey, to eat, to move when you need to actually get to a new environment that can sustain you in case the environment that you're in right now can't. So we can also pursue prey. We can also run away from predators. Okay, so this is something that animals do. Plant cells are sessile. Remember sessile that we talked about, I believe, in the first session? All right. Sessile means that generally the organism just stays in one place. It stays in one place, it doesn't move. Okay, so plant cells have cell walls because they don't need to move. So what does the cell wall do? Okay, you have the primary cell wall, and the primary cell wall is mainly made of cellulose. Okay, now cellulose, remember what cellulose is? It is actually a component of a plant. Remember what kind of a macromolecule cellulose is made of? Okay, so here we have some nice lettuce, okay, Manoa lettuce grown by my dad. He and my mom gave me some lettuce and other vegetables from their vegetable garden yesterday. You have a nice, healthy looking head of Manoa lettuce, yeah? So what are the things you see about this? For one thing, of course, it's green, yeah? So it has chlorophyll in there, and these, of course, are lettuce leaves, yeah, that actually do photosynthesis. Now, another thing that you might see about it too, that, well, you can't really see it, but you know it from when you're eating, is that, plant materials, okay, plant materials will supply you in your diet with fiber, okay? I believe they have soluble fiber or they have insoluble fiber. Both are very important for your diet, okay? So here, okay, generally when you eat lettuce, when you eat cabbage, when you eat spinach, when you eat carrots, okay, generally they say eating vegetables or fruit are very good for your diet because, of course, it has something called fiber. So the cellulose making up the cell wall of plants is actually fiber that we take in. Very important. So one of the things for your diet and for your health that you might want to do, okay, is make sure you eat a lot of fruits and vegetables. Okay, because what that does, it provides fiber, because we humans, we can't actually eat, we can't digest cellulose, okay? So we have things like termites, and we have other sorts of living things, other kinds of animals, termites, and there are cows and goats and sheep. What do? They can't actually digest cellulose either, but they have bacteria that live in their gut that helps break down the cellulose. So do you know, let's see, that generally, so cows can, of course, digest grass. So there is actually a Far Side cartoon that shows these cows, in a pasture. One of them has this outraged look on her face, and she says, "Grass? You mean we've been eating grass all this time?" So that's one thing that you can remember. Cellulose is something that we humans cannot digest. So what do? We pass it out of our body as fiber. But fiber is also very important for us to eat. Okay, make sure that we actually get enough fiber so that we can actually push things through, because the longer something sits in your gut, the more chances are that you'll get mutations in there, and that's not good. Okay, generally mutations in your body cell is not good. Okay, so just remember that. So it's important to eat your fruits and vegetables, just like your parents and other people have probably told you your whole life. Okay, so this cellulose is generally embedded in something called polysaccharides. Okay, remember polysaccharide? That's another kind of carbohydrate. So cellulose, you remember, is a carbohydrate, and the polysaccharide is also a carbohydrate too. So the cellulose fibrils that help make up the plant cell wall. Okay, if you kind of look in your textbook, okay, we'll actually see that there is an image, okay, of electron micrograph of all things, okay, an SEM, that shows a cell wall. And generally the cell wall materials, the cellulose, are laid down perpendicularly to each other, which is one of the reasons why the cell wall is as strong as it is, because what does the cell wall do? Okay, it actually functions in keeping the cell up, okay, keeping the cell up and keeping the plant erect, okay, and keeping everything going, all right? So primary cell wall is made mostly of cellulose, okay? And this particular substance is made very early in the cell's life. It actually happens right after the cell, okay, actually becomes. Okay, so we'll talk about that more in our next session, where we're going to be talking about mitosis, okay, and cell division, all right, or cell reproduction. So here, okay, in this in this drawing, generally you see here these green shaded regions of this plant cell. So this is like a cutaway, okay, showing what's inside of the plant cell. So we have here the cell walls, okay. So the cell wall of the plant here, okay, it's colored in green. That doesn't mean the cell wall is necessarily always green, okay, but generally here you see that the cell wall is exterior, okay, it's the most outermost part of the plant cell. Okay, so here, okay, so this is showing actually only a primary cell wall. Before I thought it was actually primary, this dark green, and the secondary, the light green. But I can see now, since we're looking at a cutaway, this is showing just a primary cell wall. So the primary cell wall is made very early in the life of the cell. Okay, it happens right after mitosis, okay, or cell division, okay, nuclear division and cell division, right? So here you have the cell wall, the most outermost part of a plant cell. Okay, and what does it do? It actually helps to support the plant. Okay, now we also have, in some plants, you'll have the secondary cell wall. And with the secondary cell wall, this is a component that is laid down after the cell becomes a cell. So this secondary cell wall doesn't occur in all plant cells, but generally, okay, the secondary cell wall will occur in some plant cells, including what we call sclerenchyma tissue. Okay, so the secondary cell wall has the same purpose as the primary cell wall in keeping the cell rigid, keeping the cell and the plant, you know, erect, okay, or standing up, right? So the secondary cell wall, okay, is also made partly of cellulose, but also is made of lignin. Lignin is a plant substance that is secreted. It helps the cell wall become very rigid, very, very strong, okay? So these secondary cell walls generally occur only in cells such as sclerenchyma cells, which we'll get into later. And these are cells that are specialized for support, to kind of keep the plant standing up. It also functions in protecting the plant as well as xylem cells, which are used to conduct water in the plant cell. Okay? So here, again, we have the cell wall. Okay? So the cell wall is a non-living part of the cell. Okay, the cell wall is not living, right? And the cell wall encloses the living part of the cell, which is called the protoplast. So what is a protoplast? The protoplast is the cell membrane and everything interior to the cell membrane. Okay, so everything inside or interior to the cell wall, okay, is the protoplast. Okay. Now another thing that we saw is that generally you have structures that connect one plant cell to the next called pits, right? Through these pits, materials can move from one cell into another. They will actually move through adjacent cell walls from one cell to another. And the cytoplasmic, or the connections, you know, cyto just means cell, plasmic has to do with the material, the liquid type of material that will move through these structures known as plasmodesmata. So the plasmodesmata is plural for plasmodesma. And so what these are just connections between one cell and another that helps to link them. Okay? Another structure that occurs between cells is between one cell wall and another cell wall, okay, of another cell. You'll have a structure known as middle lamella. Okay? The little middle lamella is made up of pectins. Do you know what pectins are? It's sticky, so it helps to actually stick two adjacent cells together. Okay? So generally the middle lamella, okay, occurs between cell walls of adjacent cells. So generally here, okay, we have the cell wall of the cell we've been looking at so far. And then here is an adjacent cell. Okay, right between one cell and another, you'll have the structure known as the middle lamella. Okay, it's like a sticky substance that helps the plant stick together. So you might think, okay, what is this substance called pectin? Okay, pectin, okay, so people have used pectin, okay, to do preserves. You know, like when you're making guava jelly or strawberry guava jelly or jaboticaba, any kind of fruit sort of jelly or jam that you might use, one of the things they put in there is pectin. And so pectin actually makes jelly gel, makes it sticky, makes it actually form, you know, that sort of structure that it does, you know, when you're opening a new jar of guava jelly, and then it makes it kind of sticky and makes it kind of gelatinous in a way too. Okay, so pectin is used in making preserves. Yeah, blackberry preserves, raspberry preserves, pretty much anything that you can think of. Very good, especially when you're eating it with toast or something, or peanut butter. Yeah. Okay, next then. Okay, so the protoplasm, remember, is enclosed by the cell wall. Okay, and the protoplasm can be divided into two parts. You have the nucleus being one part that we showed you a little bit earlier, and you also have the cytoplasm. Okay, the cytoplasm is the sort of like the liquidy material inside of the cell, and the cytoplasm generally is comprised of organelles. What are organelles? Organelles literally means little organs, and just like our human body, okay, with our organ systems and our organs, okay, the organelles in a plant cell actually allow for a division of labor inside of the plant cell. Okay, so we have organelles, which we are, of course, going to go into, including the chloroplasts, okay, where photosynthesis happens. You also have mitochondria, where a good part of cellular respiration happens. You also have, let's see, the ER, or the endoplasmic reticulum, okay, where proteins are made, as well as lipids, depending whether you have rough ER or smooth ER. You also have the Golgi apparatus, also known as dictyosomes, and their components that are made by the cell are finished and refined and packaged for export if it's going to leave the cell. Okay, so those are some of the organelles that we're going to cover. All right. We also have the cell sap, okay, also known as cytosol. It's actually the liquid in which these different kinds of organelles are suspended, and they kind of move around in the cell as well, okay, where they're needed. Another thing about the plant cell, okay, so the protoplasm is enclosed by the cell wall, and the outermost part of the protoplasm is called the cell membrane, also known as the plasma membrane. All right, so this cell membrane, remember what kind of organic compound is mostly comprising membranes? Do you remember what they're called? Okay, so generally you have substances known as lipids. Okay, lipids, and specifically making up the membrane, okay, of the plant cell, something called phospholipids, right? Okay, phospholipids. Remember phospholipids? They have the polar head, symbolized by my hand here, the nonpolar tails symbolized by my fingers, and they're oriented in like a lipid bilayer. You have a whole bunch of phospholipids all kind of arrayed in this way, with the nonpolar tails turned inwards, the polar heads turned outwards into the cell and out of the cell. Okay, so generally that's how membranes are structured. If you're looking at your textbook, you can follow along, okay? And in your textbook, we actually have an illustration on figure 2.6 on page 24 showing a phospholipid bilayer, okay? Phospholipid bilayer. And so what makes up the cell membranes? You generally have the phospholipids, a whole bunch of them, okay, thousands and thousands of them, with proteins embedded in the membranes. Sometimes the proteins can poke out of both sides of the membranes. Okay, sometimes only one part is poking out. Sometimes they're in the membranes themselves. So most of what makes up a cell membrane are the phospholipids and also proteins. And so one of the things, okay, is that generally, so plasma membrane, okay, what we're talking about, you get phospholipids and proteins making up the structure of the plasma membrane. And one way that people talk about it is they call it the fluid mosaic model. Now, why do they call it a fluid mosaic? They call it a mosaic because it's made up of many components. Yeah, you have all those thousands and thousands of phospholipids, and you have hundreds and hundreds of proteins in there. Okay, so what it is people think it's almost like a layer of oil, like a film of oil over the inside of the cell. So what actually helps it maintain its structure is that outside of the cell, as well as inside of the cell, you have an aqueous solution, or water. Remember that water molecules are polar. Remember that the phospholipid heads are polar also. So what happens is the heads would tend to orient themselves outside and inside of the cell, okay, with the nonpolar tails inwards, yes, in this way. Okay, so they call it a fluid mosaic because of phospholipids as well as the proteins. It can actually move around. It would be almost as if, Okay, we almost as if you go, if you were to say that the walls of your home were like the membrane, the cell membrane, you might almost like you might, okay, come home at night, go, take a shower, eat dinner, go to sleep, and then when you wake up in the morning, the front door has moved, to another room. Because a fluid mosaic model, the phospholipids can actually move. The proteins embedded in it, they can actually turn over, they can rotate, they can drift around in this membrane, okay? So another model that people use as well, they call it protein icebergs in a sea of lipids. You have these proteins, some of it sticking out, okay, some of it, floating around in this membrane. So you actually have these proteins, a good part of them embedded in the membrane. So another model people have quoted is calling them protein iceberg in a sea of lipids, because the lipids can actually move around amazingly. Okay, so this is the model that most people follow nowadays when they're talking about the cell membrane. Okay. And another thing about the cell membrane, very important, is that it has a function. What does the cell membrane do? It controls what can get into the cell, okay? It controls what can go out of the cell, okay? And etc. Okay, so this is the main function of the plasma membrane. Another thing about membranes is that almost all organelles inside of the plant cell have membranes around them. They're either membranous organelles, okay, or they have membranes around them. Okay, so membranes are very important in terms of what can get into a cell, what can leave a cell, what can get into a nucleus, what can leave a nucleus, and etc. Okay, so the cell that we're talking about now is extremely complex and very, very fascinating. Okay, and remember what particular subdiscipline of botany studies cells? Well, you have cytologists for one thing, but remember plant anatomists. Plant anatomists study cells and they study tissues inside of plants. Okay, remember that? Yes. Okay, now generally too, another component of the protoplast are organelles. Okay, what are organelles? Remember, organelles are like little organs inside of a cell. Okay, and one organelle that we're going to talk about, this will be one of the last things we talk about today. One kind of organelle is called a chloroplast. Okay, chloroplast. Okay, a chloroplast is a kind of plastid. Okay, and generally today we're going to talk about 3 different types of plastids. And the primary one we're going to talk about, of course, is the chloroplast. So when you think about the word itself, chloro, when you think of Clorox or you think of chlorine, right, you think of like a kind of like a yellowish green color, right? So chloroplast. Plastids are bodies inside, and these little bodies or little organs, these are the organelles in which photosynthesis happens. Very, very important, as we talked about before, to life on Earth. Okay, chloroplasts, these are the organelles of photosynthesis. Okay? Now the chloroplasts are surrounded by the outer part of them, which are called a double membrane. So chloroplasts have a double membrane around them, and they do the world's most important biochemical process. They do the process known as photosynthesis. Recall that photosynthesis is responsible for all the food that we eat. All the food that we eat basically comes from photosynthesis when you trace it back far enough. Remember, you either have plants that we eat or we eat animals that eat the plants. Okay, either way, in fact, both of them. Okay, so chloroplasts is where photosynthesis happens. Very, very important. Okay. Another thing about the chloroplasts is generally they contain pigments. Okay, what are pigments? Pigments are substances that lend color, okay, to this particular structure and lend color to the plant. Recall that when we were looking at that head of lettuce, okay, that we had chlorophyll, which is the pigment. So one kind of pigment, okay, in a plant are called chlorophylls. Generally, land plants have chlorophylls A and B. Okay, remember we talked about this before? One is a yellowish green pigment, the other is a blue-green pigment. And because they're a little bit different in color, they can absorb a little bit different parts of light spectra. Yeah, because the visible light goes all the way from red, okay, into orange, into yellow, into green, okay, different kinds of greens, into blue, and into the violet. Okay, so pigments, okay, very important. So one kind of pigment that generally plants have are chlorophylls. Okay, chlorophylls A and B. All right, very important. okay? Chlorophylls are the main type of pigment. Another kind of pigment that plants have, okay, are called carotenes, all right? Carotenoids, okay? And the carotenes, okay, are of different types, all right? And among them we have xanthophylls. So chlorophylls generally are greenish. When we look at chlorophyll, okay, in a plant, generally they're green in colors, okay? When you look at carotenes, they generally tend to be orange or red. Okay, they help the plant absorb in different parts of the spectrum. Then we have xanthophylls. Xanthophylls tend to be, I guess, yellowish and also maybe brownish. So when you're actually looking at leaves, usually what you'll see is a green leaf, okay? So generally, okay, when you also, in places such as New England, places that are temperate in climate, what you'll usually see is that in the fall, okay, one of I guess in temperate areas, so, the best times of years to visit, of course, is springtime when the flowers are all coming out, and also in the fall when the leaves are turning color. They call it that the leaves are turning color. What's happening then is that the leaves are actually senescing, they're dying. What then happens is that the chlorophyll pigments will break down, and then the browns and the yellows and the oranges that are actually in there due to the presence of carotenes and xanthophylls, that's when you see these fall colors, because what happens is the chlorophylls have faded and they're dying, and what happens is that the other pigments are then exposed. That's why you can see these beautiful reds and oranges and yellows and browns, in the aspens and the oaks and all these different kinds of beautiful deciduous trees in the deciduous forests, yeah, like places on the mainland, yes. Okay, so primarily, chloroplasts are the sites of photosynthesis. That's always the most important thing to remember about chloroplasts and plants in general, okay? And so what are they comprised of, okay? They have two parts that are important to us, okay? One part is called the grana. That's a plural form of it. The singular is granum, G-R-A-N-U-M, okay? What are grana? Grana are stacks of membranes known as thylakoids. Okay, so the grana is one part of the chloroplast, and the grana are the places where the first part of photosynthesis, known as light reactions, happen. The other part of the chloroplast that need to concern us is called the stroma. Okay, don't confuse this with stoma, okay, because stroma is like the jelly-like substance inside of the chloroplast, and the stroma is a place where the second part of photosynthesis happens, and that part is called the Calvin cycle. Okay, so the first part of photosynthesis is known as the light reactions, okay, because of course there's more than one reaction that happens during the light reactions. The other part of photosynthesis is known as the Calvin cycle. So in the grana, you have the light reactions happening. In the stroma, you have the Calvin cycle happening, okay? So here we have a beautiful image, okay, of a chloroplast. So here you can see that the chloroplast has an outer membrane, right? You also have an inner membrane, okay, of the chloroplast, okay? Outer membrane and inner membrane. So a chloroplast has a double membrane around it, okay? In here, each of these kind of coin-shaped structures here is known as a thylakoid. Okay, as you can see here from this word, yeah, thylakoid. And these thylakoids are made of membranes. There are actually a bunch of membranes in here, inside of this organelle. So you have these stacks of thylakoids, and they actually make up the substance, the structure known as a granum. Okay, so here you have a single granum here, and here you have multiple grana, okay, making up these structures. So the grana are places where the light reactions happen. So the light reactions happen on the grana, okay, in the thylakoid membranes, because in the membranes you have embedded in the membranes you have structures known as photosystems. Okay, we're going to get into that when we get into photosynthesis, okay, which is going to be chapter 4. All right, so we won't come to that for a while yet, but like a preview, okay, the light reactions happen in the thylakoid membranes that comprise the grana. The light reactions happen in this gelatinous substance. So inside of a chloroplast, you have this like a jelly-like substances, and this is called the stroma. Okay, so inside of the stroma, that's where the Calvin cycle happens. Okay, inside here. So we have the light reactions happening in the grana. We have the Calvin cycle happening in the stroma of the chloroplast. Okay. So you can see that even something like an organelle here, that even a tiny little organelle, remember, so an organelle is kind of in the cytoplasm, even the organelle is very complicated. Okay, so isn't it amazing? We can actually view and look at images of chloroplasts as well, okay, when we're looking at photomicrographs, especially scanning electron micrographs and transmission electron micrographs, okay? Now we have other kinds of plastids. Remember that chloroplasts are a kind of organelle that is sort of classified as a plastid. Other kinds of organelles include the leucoplasts. Okay, leucoplast, leuco refers to white. Remember that chloro refers to a green color. Leuco means white, such as leukemia. Unfortunately, we have this unfortunate sort of connotation or basic root. So leukemia is a cancer of white blood cells. Either the body produces too many white blood cells, which crowd out the erythrocytes or the red blood cells, or you have abnormal, right, white blood cells being produced. So leuco means white. Okay, so leucoplasts, what is their function? Here we have a nice image of some leucoplasts. They actually are starch grains. They will actually store starch inside of them. So this is probably a cell of potato, white potato, yes, Solanum tuberosum. So you can see here the cell walls, okay, of this particular cell. You can see it right here at the edges. And then you have the leucoplasts with the starch grains in here. Okay, usually starch will stain either black or purple or brown or blue if you add potassium iodine to it. So the person who made this slide probably stained it with IKI. Another kind of organelle or another kind of plastid is called a chromoplast. Okay, a chromoplast is a kind of plastid that I'm sorry, not a plastid, but a plastid. Okay, a plastid that will they will actually account for something like this. Here we have a little, okay, example of Widelia, okay, or Widelia trilobata. And this here, you can see here, it looks like a flower, doesn't it? Okay, but anyway, getting back to the subject, here we have an image of a flowering sort of organ, okay, having this yellow coloration. So oftentimes in flowers, okay, as well as in fruits, sometimes the chromoplasts are responsible for the pigmentation that we see in yellow or in orange, okay, fruits, or in such as tomatoes, because tomato is actually a fruit, amazingly, okay. Tomato and bell pepper, you know, when they're yellow or they have ripened to yellow, or if they've ripened to orange, or if they have ripened to red, they will have chromoplasts in them. Okay, so the chromoplast can be responsible for this kind of coloration, okay, inside of a plant cell. All right. Anyway, so let's see. So let's continue a little bit, okay, just a little bit, okay. Another kind of organelle, very important, okay, not only important in plant cells, but also important in animal cells as well as fungi, pretty much, okay, any kind of cells that we call eukaryotic cells, and these are called mitochondria, okay. Mitochondria are plural. Mitochondrion is singular, okay. The mitochondrion is where cellular respiration takes place, or parts of cellular respiration take place, okay. So mitochondria, similar to the chloroplast, is surrounded by a double membrane, okay? And the inner membrane of the mitochondrion has these infoldings, okay? Also known as cristae, right? The inner membrane has all these infoldings, and why do they have these infoldings? It's because the inner membrane has these structures in them called electron transport systems. All right, and if you have a larger, you know, kind of folded inner membrane, that increases the surface area of the inner membrane, so you can have more and more electron transport systems inside, on top of the membrane, or on top of, inside of the membrane. Another part of the mitochondrion, which is important, is called the matrix of the mitochondrion. Okay, so generally, so we talked about photosynthesis having two parts of it, including the light reactions and the Calvin cycle. Well, cellular respiration, Okay, has three parts to it. We humans divide it into three parts. One part is called glycolysis. Glycolysis generally happens in the cytoplasm. Okay? The next part of cellular respiration is called Krebs cycle, okay, named after Hans Krebs. And the Krebs cycle happens here, inside of the matrix, in the inner matrix of the mitochondrion, okay? So we have glycolysis happening in the cytoplasm. We have the Krebs cycle happening inside of the matrix of the mitochondrion. And the last part, which is called the electron transport chain, happens in these membranes here. Okay, so here you have the outer membrane of the mitochondria, and then you have these infolded cristae. Okay, they make up the inner membrane. And the inner membrane, you know, has these infoldings due to the fact that it increases the surface area of the inner membrane. The more surface area you have, the more electron transport systems you can actually have on these membranes. Okay? So respiration is extremely important in plants and in animals, because what does cellular respiration do? It actually will take glucose and other kinds of organic compounds and break it down into a form of energy that the plant and also us animals can recognize, which is called ATP. Okay? So generally inside of the inner, on the inner membrane of the mitochondrion, you'll have also structures, right, called electron transport systems, but you'll also have structures known as ATP synthases. Okay? ATP synthesis will actually make ATP. Remember we talked about ATP maybe a little before? ATP is what people call the currency of the cell. It is a form of energy that the plant can actually recognize and use to make more plant, okay, or to power forward the various kinds of biochemical events happening inside of the plant. Okay, so these ATP synthases are enzymes that produce ATP. Okay, very important. So the mitochondrion, the main function of the mitochondrion is to do cellular respiration. Okay, so that's a function of the mitochondrion. Okay. Right. Okay, let's see. So in today's lecture, okay, we talked about some various things. We talked about the cell, okay, and how it is a basic unit of life, okay. We talked about the cell theory and about some of the early workers. Remember, the cell theory has to do with, right, that all living things are comprised of cells. The next thing about it is that generally all living things, okay, come from other living things. Cells can only arise from other cells. You can't actually have a cell coming from something that's non-living. So life comes from life. Okay, you can't actually have a living thing coming from something that is non-living, as far as we know so far. Okay, so generally in science we tend to just go with what we know. Another thing that we talked about the early users of the microscope, including Robert Hooke and including Schleiden and Schwann, who formulated what we now know as the cell theory. All right. We also talked about the compound light microscope. We also talked about electron microscopes, right? The SEM, or scanning electron microscope, and the TEM, also known as a transmission electron microscope. Okay? Furthermore, we started talking about the components of the plant cell. Yes? We talked about the outermost part of the plant cell, which is known as the cell wall. We have found out that you can have a primary cell wall. You can also have a secondary cell wall in some cells, especially in cells that are specialized for support, okay, and for water transport, for kind of keeping the plant up, you know, so lending support to the plant. All right, so we talked about the cell wall with its cellulose fibrils laid perpendicular to each other. Okay, that accounts for the strength of the cell wall. So the cell wall provides for exterior or external support for the cell to keep the cell rigid. Okay. Another structure that we talked about the cell membrane, which is the outermost part of the protoplast, or the living part of the cell. Because recall that the cell wall, though it's an important part of the cell, is non-living. It is not actually alive, even though it is produced by the living part of the cell. Recall too that, so the cell membrane, what does it do? Remember? Think about it. Okay? The cell membrane controls what can get into the cell, what can exit the cell, what can enter the cell, and what can leave. Okay? So very, very important, because the cell membrane in many organisms forms the boundary of the cell. But of course, in plant cells, you have the cell wall. Interior to that, you have the cell membrane. Okay? Another thing we talked about a little bit about the cytosol and also about the organelles inside of the cell. Okay? So generally, so you have here the organelles of the cell, right? And the cytoplasm, or the cytosol, which is the liquid inside of it. So one of the things that we talked about, yes, was, let's see, we talked about the chloroplast, okay? So the chloroplast, of course, is the organelle that is responsible for photosynthesis. Recall that the chloroplast is also made of several components. It has a double membrane around it. It has two membranes surrounding the inner part of it. And inside of the chloroplast, you have stacks of thylakoid membranes. And what are they called? Remember what they're called? Okay, they're called grana, remember? And then when you actually figure out the function, or we humans have figured out the function, okay, of the grana, what does it do? Okay, in photosynthesis. It does the first part of photosynthesis, also known as the light reactions. Okay, we're going to go into that later, but the light reactions, of course, take place only when there's light. Okay, so the light reactions have to do with the harvesting of light energy, converting it into chemical energy. Okay, now another part, okay, of the chloroplast, okay, of course, is known as the stroma, S-T-R-O-M-A. Okay? The stroma is a part of the chloroplast. It's like a jelly-like matrix. There's like a lot of proteins, a lot of enzymes, a lot of chemicals in there. And what does the stroma do? What happens in the stroma? Do you remember? Okay. It's called the Calvin cycle, right? So the Calvin cycle happens inside of the stroma. Okay, so we have the light reactions happening in the grana, and you have the Calvin cycle that happens in the stroma. So generally in photosynthesis, you have two parts. Next, we talked about the mitochondrion. Okay, remember the mitochondrion? What does it do? It does another biochemical process known as respiration. Recall that respiration has three parts to them. Okay, this is like a little introduction to it, so please don't get worried about it yet. Okay, we're going to cover this when we get into chapter 4, but I wanted to give you a little preview, right? So generally, respiration happens in three parts. You have glycolysis that happens in the cytoplasm. Okay. Secondly, we have the Krebs cycle that happens, okay, in the matrix, in the innermost compartment of the mitochondrion, right? And then the third part of cellular respiration is known as the electron transport system. Okay, there's a whole bunch of different electron transport systems all embedded in the cristae, okay, of the inner mitochondrial membrane. Okay, all right, let's see. Okay, so I'm glad that you folks stopped in. Okay, another thing I wanted to tell you is that if ever you have any questions about the subject material that we're covering, you can email me anytime. Okay, my email address is on the syllabus. All right, so this is what we have wanted to cover today, and I'm glad that you're here. I'm glad I'm here too, and we'll see you next time. Okay, bye-bye.